Cells, the structural and functional units of all living organisms. Definition of a cell. A cell is the smallest unit capable of independent life in all vital physiological processes, such as metabolism, growth, reproduction, and response to stimuli. Every organism, unicellular or multicellular, is composed of one or more cells. Hence, cells are both the building blocks and the functional units of life. Structural role of cells. Cells form the physical structure of all living organisms. They are the foundation upon which tissues, organs, and systems are built in multicellular organisms. Examples: muscle cells form muscle tissue. Nerve cells form neural networks. Epithelial cells form skin and linings. In unicellular organisms, the cell itself performs all life functions. Functional role of cells. Each cell performs essential life functions, such as energy production, ATP generation, biosynthesis, proteins, nucleic acids, lipids, information storage and transfer, DNA, RNA, transport, active, passive transport of molecules, response to environment, signal transduction, growth and division, mitosis, meiosis in eukaryotes, binary fission in prokaryotes. These processes are carried out in specialized compartments, organelles, in eukaryotic cells and in a more general cytoplasmic environment in prokaryotes. Cell types. 1. Prokaryotic cells. Examples: bacteria, archaea. Small, simple. No nucleus. DNA in a circular form. No membrane-bound organelles. Reproduce by binary fission. 2. Eukaryotic cells. Examples: animals, plants, fungi, protists. Larger, complex. True nucleus. DNA in linear chromosomes. Contain organelles, mitochondria, Golgi, lysosomes, etc. Divide by mitosis, meiosis. Cell theory. Modern form. All living organisms are made of one or more cells. The cell is the basic unit of life in all living things. All cells arise from pre-existing cells. Energy flow, metabolism, and biochemistry occurs within cells. Cells contain genetic material, DNA, passed from one generation to another. All cells are basically the same in chemical composition. Experimental support and observations. Microscopy, light and electron, direct visualization of cells. Cell fractionation, demonstrates compartmentalization of function. Radioisotope tracing, follows metabolic pathways in cells. Molecular biology, confirms DNA replication and gene expression at the cellular level. Unity and diversity of cells. Despite immense diversity among organisms, all cells share a common biochemical machinery: DNA, RNA, proteins. Energy conversion processes. Examples: glycolysis, TCA cycle. Similar membrane structures. Ribosomes for protein synthesis. This points to a common ancestral cell, the last universal common ancestor, LUCA. Summary points. Cells are the smallest units of structure and function in life. All life processes occur at the cellular level. Cells can exist as single-celled organisms or as part of a complex multicellular structure. The organization, biochemistry, and genetic machinery of all cells are fundamentally similar. There are 3 distinct domains of life. Overview. All living organisms can be classified into 3 domains of life, based on molecular phylogeny, especially ribosomal RNA sequencing, biochemical and genetic characteristics. The 3 domains: bacteria, archaea, eukarya. This classification system was proposed by Carl Woese in the 1990s using 16S and 18S rRNA sequencing. Bacteria. Eubacteria. Characteristics: unicellular, prokaryotic organisms. Lack nucleus and membrane-bound organelles. Cell walls usually contain peptidoglycan. Circular DNA in a nucleoid. Ribosomes are 70s. Asexual reproduction. Binary fission. Examples: Escherichia coli model organism, Bacillus, Streptococcus, Cyanobacteria. Photosynthetic bacteria. Biochemical features: use glycolysis, TCA cycle. Oxidative phosphorylation can be aerobic or anaerobic. Great metabolic diversity: fermenters, photosynthesizers, nitrogen fixers. Archaea. Characteristics: also prokaryotic, but genetically distinct from bacteria. No peptidoglycan in cell wall, instead use pseudopeptidoglycan. Unique membrane lipids with ether linkages. Ribosomes are 70s, but structure resembles eukaryotic ribosomes. Extremophiles, thrive in high salt, temperature, acid. Halophiles, thermophiles, acidophiles. Examples: Halobacterium salt-loving. Thermoplasma, methanogens. Methane producers. Biochemical features: distinct enzymes for transcription and translation closer to eukaryotes. Unique pathways for energy production. Examples: methanogenesis. Can be anaerobic or aerobic. Eukarya. Eukaryotes. Characteristics: nucleus enclosed by a nuclear membrane. Membrane-bound organelles. Examples: mitochondria, Golgi. Linear DNA organized into chromosomes. Ribosomes are 80s. Can be unicellular or multicellular. Includes animals, plants, fungi, protists. Biochemical features: complex regulation of gene expression. Compartmentalized metabolism. Mitochondria and chloroplasts have their own DNA supporting endosymbiotic theory. Phylogenetic tree based on rRNA. Ribosomal RNA sequences are highly conserved. Carl Woese's analysis of 16s18s rRNA led to the 3 domains model. The tree of life shows bacteria branched early. Archaea and eukarya share a common ancestor. They are more closely related to each other than to bacteria. Luca. Last universal common ancestor. The root of the 3 domains traces back to Luca. Luca was likely a simple, single-celled organism with DNA-based genetic system. RNA intermediates. Ribosomes. A membrane. Key takeaways: life is classified into 3 domains based on molecular phylogeny. Archaea and eukarya are closer to each other than to bacteria. Understanding domain differences is essential for molecular biology, genetics, and evolutionary biochemistry.